So welcome back to the BtBsDay edition 2020. I'm very pleased to introduce to you Antonella Ronchi, who will present Nicole Soranzo, our next speaker.
Okay, so welcome everybody, welcome Nicole. It is a real pleasure and also an honor for me to introduce you our keynote today's speaker, Professor Nicole Soranzo. Nicole is a human geneticist and is one of the major experts in the field of the study of human complex traits at the genomic level. Nicole graduated in Milano, but then she moved for her PhD at the University of Dundee, and she took a PhD in genetics. She then moved to the University College of London, where she started to work on the methodological approaches to study evolutionary genomics and genome-wide association studies. She then went to US at the pharmacogenomics department at the Johnson & Johnson. And after 2 years, she went back to UK, to Cambridge, when she started her own lab at the Wellcome Trust Sanger Institute. In 2015, she was appointed Professor of Human Genetics at the School of Clinical Medicine at the University of Cambridge. And since then, she's part as a leader with the leadership position in many groups, in particular in groups interested in the study of platelet biology and cardiovascular genomics. So she's part of several steering committees and scientific advisory boards, but I just would like to mention that she recently became a fellow of the Academy of Medical Science and of the European Molecular Biology Organization. And I'm particularly happy to announce that starting from next year, she will be back in Milano at the Human Technopole as a joint head of the Center of Genomics. So Nicola, I apologize because for sure I didn't mention part of your many awards and achievement, but I didn't want to take too much time to your seminar. So thank you very much for being here today. And let's start your seminar.
So, prima di tutto grazie a tutti, veramente un piacere essere qui a parlare con tutti voi. Sono via dall'Italia da quasi 20 anni e il mio italiano è un po' arrugginito, per cui parlerò in inglese, ma è veramente un grosso piacere rincontrare la comunità milanese, in particolare da cui provengo. So what I would like to do today is to give you a little bit of a general overview about some of the studies that we've been doing in the last 15 to 20 years to try and understand or unravel the genetic basis of human traits and diseases. And what I would really like to get you away from this is, first of all, the sense of the speed of transformation that we have experienced in the field, but also the potential opportunities for understanding and treating human diseases. So I think I don't really need to say too much about the importance of studying common diseases. Common diseases are indeed the biggest causes of not only mortality, but also morbidity in the human world. And so cardiovascular disease alone accounts for about 15% of the death worldwide, and results in about 365 million disability adjusted years worldwide. So these are, it is really important that we become better at both understanding the causes of disease, but also obviously treating them. The other consideration is that common diseases, in particular non-communicable diseases such as obesity, cardiovascular disease, and many others, are on the rise. And this is certainly due to the change in environmental conditions that are increasing morbidity, particularly in developing countries. And as you see on this plot on the right, these diseases really don't spare anybody. There is a fairly stable risk of disease and across the world, with minor differences linked to local conditions. So this is a problem that affects everybody. And we really know a lot about disease, and we can track disease not only with disease itself, but also by tracking what are the most important disease factors that we need to tackle. And so, for instance, if you see high blood pressure alone accounts for over 10 million deaths worldwide. So we can gain insights about disease risk not only by studying disease, but indeed by a much deeper understanding of the underlying disease factors. So what I would like to tell you today is a little bit is how can we use analysis of DNA to help tackle some of the big challenges in human health. As I mentioned before, human complex diseases arise from a combination of your underlying genetic predisposition and in conjunction with environment, and we really only focus on the first part, so on the genetic predisposition. And there are really three fundamental challenges here. One is around discoveries, on mapping genetic variation. Is really the challenge here is to understand which genetic variants affect human traits, disease risk, and patient outcomes in different populations. The second big challenge is through which molecular mechanisms do these genetic variants act, and this particular challenge identify what are the causal mechanisms underlying some of these associations, because those are the ones that we can act on to modify the risk of disease. And the third big challenge is around translation. In particular, how can we harness the discoveries that we made in human genetic studies to improve the prognosis, diagnosis, and treatment of disease. And so I'm going to take you through some of these challenges and how we've been addressing them. So starting with the genetic discoveries, what's really propelled the, sorry, I need to remove a small window. Okay, so what's really propelled us in our research has been a combination of technological advancements, but also the generation of large-scale resources for genomics. And so if you're interested in this topic, there is a review that we wrote together with many colleagues worldwide that really summarizes the last 15 to 20 years of human genetic discoveries, both in common disease, but also in rare disease. And as you see, obviously, one of the main discovery was the publishing of the draft human genome, but it did also the progressive realization that genetic variation comes in recognizable structure, what we call haplotype, and we can exploit this haplotype structure to obtain characterization of genetic variation genome-wide. And so as you see in these timelines on the left for rare disease and on the right for common disease, over the course of the last 20 years or so, we have certainly gained a lot of understanding about the cause of genes mechanism underlying rare monogenic diseases, but more recently also had an exponential increase in the number of disease predisposing variants for common disease that we have identified. And Sanger is certainly one of the places in the world where these large-scale genomics studies have been conducted. So the approach that we use to study or to discover genetic variants is through effectively analysis of human populations. And this is really one of the type of designs that we use, what we call population-based cohorts or large-scale biobanks. There are others. And in this particular case, we really harness the fact that we have a very large collection of healthy volunteers. So this is a plot from the UK Biobank, which includes 500,000 people, where participants have been selected to be predominantly healthy with some biases and have a population representative of the whole population. Typically, these participants are very widely phenotyped. They have extensive phenotypes, thing that doesn't only include risk factors for disease, but also an increasing number of candidate biomarkers, imaging, and other types of omic data. The power of this type of studies is that we have very good understanding of both environmental risk. This comes from questionnaires, physical activity, objective measurements, as well as neurocognitive measurement. And progressively also more and more the ability to track disease over time through linkage to electronic health records, self-report, and hospital discharge records. So what we do is effectively characterize genome-wide genetic variation in these individuals through a number of approaches, and then to test through association studies the correlation between genetic variation and disease risk. And from a purely analytical point of view, this has a lot of advantages. We can use this as genome-wide unbiased discovery approaches, where effectively we look through the genome without any prior assumption about the biology that underlines a certain trait. They can be very cost-effective and also statistically powerful because we do a single genetic experiment, and then we can test association with hundreds, if not thousands, of different phenotypes in the same person. But importantly, perhaps this is less realized, is that the analysis in population representative or healthy individuals are useful because they, in these people, the correlation between a phenotype and a genotype is not affected in a systematic way by environment, what we call confounding, or disease state, what we call reverse causation. And so this provides us with much more robust ways of assessing the impact of genetic variant on a phenotype. So from an analytical point of view, as I said, the approach is relatively straightforward. We initially used single nucleotide arrays that capture hundreds of thousands of variants in tens of thousands to hundreds of thousands of people. We know now the underlying haplotype structure of the human genome, and basically we can use this sparse array to fill the gaps and obtain millions of variants for testing. And then we correlate this, as I said, with phenotype. And again, for phenotype, there are many different ways in which you can define phenotype. So this is the case of cardiovascular risk. You could define a simple case control study where you classify a person based on whether, for instance, they had a heart attack at a given point of time or not. But indeed, you can also use a much more sophisticated measurement that capture the underlying pathophysiological or molecular components of the disease. And for instance, you could look at arterial imaging to define quantitative scores, for instance, of arterial blockage, or indeed look through what we know about the underlying pathophysiology of some of these diseases and define, for instance, markers of inflammation or apoptosis that are measured quantitatively and can be used in association study. And so, so far, what we've been able to do quite relatively easily is to do studies of disease predisposition and risk factors. But there's a huge amount of research, I think, and understanding that still needs to be gained from studies of different types of patient outcomes, for instance, understanding of disease course, severity, and response of treatment. And for this, we're still very much in the early days of discoveries. So if we want to capture, perhaps in a more powerful way, the breakneck speed of the discovery, since I also started working in complex diseases, we had a first genetic variant associated with macular degeneration back in 2005. And as we move over time, we have progressively filled the human genome with genetic variants that predispose us to different diseases or risk factors. And so now we are at the stage where we have more than hundreds of thousands of these genetic loci. And having this collection of genetic variants in general has allowed us to also better capture some of what we call the allelic property of the spectrum of complex race and diseases. So how these genetic variants behave in total. And so there are several characteristics that emerge also quite clearly. The first one is this pleiotropy, where we see that a single genetic variant can be associated with multiple traits or multiple diseases. And this often reveals really interesting and important insights into either comorbidity, so genetic predisposition to, for instance, having multiple diseases, or indeed causality, where we identify, for instance, a risk factor that is responsible for a particular locus affecting risk of disease, so a disease onset. In terms of the allelic architecture, These genetic variants have been discovered through early stage GWASs, tend to be quite common in the population, and this is partly a result of the ascertainment of the original genome-wide association studies, but very compellingly, they are very individually very low or modest effects on phenotypes, which means that for every additional genetic variant that a person carries, you only at best have an increase in risk of a few percent. And so the argument is that these genetic effects individually are indeed quite negligible. The third type of observation that we made of now looking at variants systematically is that these genetic variants, for the most part, map to non-coding region of the human genomes, and it's often quite difficult to link them to their genes or indeed, for instance, regulatory elements of interest. So much of the work that I've been doing following discovery of this, or in parallel to the discovery, is to, first of all, try to understand what is the underlying effector genes and mechanisms that underlie any of these associations. And this is important because it gives us important new insights into pathophysiology of the disease that was previously undiscovered. And the second one is how best can we use or harness this genetic information in the clinic. So as I said, these genetic variants individually have effects, but what we're beginning to see as you add up the genetic variants, tens or indeed hundreds of genetic variants, you're beginning to see some resolution of phenotype. So this is a very nice paper by a colleague in Boston that showed that effectively, so you can model any phenotype. In this case is body mass index, but this is indeed applies true to nearly every disease we looked at. And indeed, you see that individuals, depending on how many copies of the allele, fall somewhere on the scale of this bell curve distribution that is BMI. And so individuals that typically, there will be few individuals that typically will carry a very small number of risk alleles across all the genetic variants we've identified, and this results in a fairly low phenotype, versus others that carry a particularly high burden of genetic variants and indeed a high phenotype or disease risk. And obviously the majority of population sits somewhere in the middle, and this is by definition. And so you can begin to use this, what we call polygenic risk scores, to stratify population based on people that at birth were assigned either a very particularly high or low risk of disease. And so, for instance, you see that if you compare people that are at the very low end of the spectrum versus those that are very high of the spectrum, you have very uneven representation. So that, for instance, people classified with obesity tend to have a particularly high burden of disease versus people that are classified as normal or indeed underweight. They tend to be enriched at the other end of the spectrum. And obviously, an interest is to what extent are these polygenic risk scores predictive, so allow us to tell us, even at the beginning of life, whether a person will or not go on to develop a given disease. And it's fair to say this is work in progress, but we can see that there are already evidences or signs that at least for people at extremes, this approach could be useful. And so this is just a way to show that polygenic risk scores capture genetic effects differentially depending on context. So, for instance, the same polygenic risk score is not very predictive in babies or young children, but becomes quite predictive later on in life. And this is another example of polygenic risk scores that allow us to stratify risk, in this case for BMI, in older versus younger people. The other really, so as I said, these polygenic risk scores are beginning to be useful. They really add up information from hundreds of genetic loci, but we definitely want to do better than that. And what the genomic field is now pushing is the discovery and more genetic variants through genome sequencing, whole genome sequencing of millions of people. And the idea here is that there are other variants that sit somewhere in the rare end of the spectrum. They tend to have low allele frequency, so they could be carried by one or few people, but have much bigger effect sizes. So beginning to approach the size of Mendelian disorder variants, but not quite there yet. And so what we are doing at the moment through studies, and these are some of the studies that my team has published, has been to scan systematically the protein coding and the non-coding regions of the genome to discover these variants, first of all, and then to test them in association studies. And because these variants are very rare, you cannot test them one by one, but you can begin to add their effect by, for instance, combining information across exons and genes. And what we're finding is that we indeed are beginning to discover quite a few of these more rare alleles. And as we were hoping, these are variants that are indeed rare, but they increase the risk by a much larger amount, the common variation. So in carriers, they are becoming more predictive compared to common variants. And indeed, they are also enriched in variants of more interpretable consequences, such as, for instance, gain or loss of function mutations. So as I said, we have now sitting on a treasure trove of hundreds of thousands of genetic variants. And so the next question is really to begin the painstaking approach of annotating each variant to the underlying causal mechanism. Because it is expected that indeed different variants may act only in different tissues and different developmental contexts, or indeed only in response to a particular stimulus, for instance. So really the basic questions here is what are the genes involved, what are the mechanisms, what are the pathways, what are the cells, what are the developmental stages or environments that each variant exerts an effect on disease on. And again, there's a nice summary of approaches and opportunities and pitfalls in this review that you could want to look at. This is just to say that while discoveries have proceeded very, very well, there's been a lot of, a lot more challenge. There's a lot more challenging the studies are very painstaking, take a lot of time, and we're not only now beginning to look at variant to function studies at scale. In order to approach linked annotation of genetic variants, there are multiple approaches, and again, technological development over time is helping in this process. There are approaches that rely on integration with the multi-omic data sets, including not only transcriptomics, but progressively more sophisticated maps of, for instance, chromatin structure and function. There are studies that map the effect of genetic variants through different types of phenotypes, and I'm going to tell you a little bit on the next slide, as well as the opportunity to then go in vitro system and engineer genomes systematically to look at the effect of these genetic variants. And finally, overlaying association with catalogs of genetic and genomic variation. So the first part, so these in vivo approaches are in fact a type of approach that my lab has been particularly active on. And so the basic idea here is that we can treat again human populations as if you want to live in laboratories, and we can use new technologies that are, when they become scalable and sufficiently cheap, so that we can profile intermediate layers of variation between the primary effect of genetic variant and its final effect on a complex phenotype or disease. And so we looked at association across a number of layers, including gene regulation phenotypes such as expression and splicing, but also methylation, structure, chromatin, and transcription factor binding. We looked at metabolic phenotypes, particularly through small molecule metabolites such as sugar, amino acids, lipids, and all intermediates of biochemical reactions. Importantly, now we have an opportunity to look at protein levels quantitatively, because that gives us an immediate readout of the gene that a given variant might impact. And we also looked extensively at cellular composition QTLs to look at, particularly in blood, how whether genetic variants might shift, for instance, a balance between different types of immune cells. As I said, the other approach, in parallel to this in vivo, are in vitro approaches, and these are becoming progressively more feasible and cost-effective. And the basic idea here is really that we want to take a genetic variant associated in a GWAS study and then map their function against different type of regulatory elements. And this is particularly important because we know 90% of our associations so far map to non-coding regions and have a predicted regulatory effect. And so there are now techniques that are becoming scalable, including exogenous techniques that look at pooled reporter assays. For instance, it can take a genetic variant or a regulatory element, and we design specific probes that bind to this region and then measure activity in vitro. And importantly, now we can also set up experiments where we indeed engineer these genetic variants in vitro and express them to derive functional relevant phenotypes such as functional assays, proliferation assays, or indeed other types of cell function assays. And really, ideally, what we want to do is to get to a point where we can target each variant in the genome, each regulatory element in each gene, and derive functional readouts that allow us to interpret the associations better. So as I said, this process of mapping mechanisms is long and painstaking, but it's really important to get to a better understanding of the mechanism and the underlying disease predisposition. And one of the motivations, in fact, strong motivation for this type of experiments is that we know very well that an understanding of genetic risk, and in this case, particularly coming from rare diseases, has really been proven instrumental in designing treatments for disease. And this is not only in designing targeted therapeutics, such as, for instance, in the case of ivacaftor for targeting CFTR for cystic fibrosis, but indeed for a much broader range of applications, such as, for instance, information that allows to treat disease or ameliorate disease by altering diet or behavior. And this is the case, for instance, of many metabolic diseases. Or to, for instance, design a prophylactic intervention or increase surveillance once we know that a particular individual is a carrier for a genetic risk factor. So the question is, how can we now design similar type of approaches also for the common disease? And again, because of the complexity of this disease with multiple, polygenic risk, and also the challenges in identifying the underlying mechanism, this becomes much more difficult. There is, however, a lot of interest at the moment in the field, and this is indeed why many pharma companies are beginning or have begun to invest systematically in genomic studies, which is that genetic analysis or genetic information can aid substantially in two fundamental bottlenecks of the drug development process. One is to aid drug discovery by identifying through in silico or genetic approaches what are the compounds that are more likely to be effective and result in successful drug development. And the second one is obviously in patient stratification, as shown earlier for the polygenic risk scores, is that we can begin to look more systematically at whether patients are more or less likely to respond to a given treatment, and therefore design treatments, clinical trials for that. So effectively, what we do in this type of approaches is that we consider genetic variation as effectively an in silico version of a drug. So we use genetic information to mimic what would be the pharmacological effect of modulating a given target through a given drug. And we seek to build those response relationships that allow us to link in silico what is the correlation between the genetic perturbation on the gene and the effect on disease. And if there is no correlation, then we know that that's not a good target for disease. But indeed, if there is correlation, we can begin to prioritize this type of drugs for treatment. and this is a very nice review if you want to, if you're interested in this topic. And indeed, there are now a number of examples where this type of approach has been effective, both to discover new drugs, but also to design trials where we can repurpose old drugs for new indications. This is obviously very efficient because the drug is likely already developed and approved and just needs to be applied in different cases. And obviously, we've seen recently in the case of COVID how powerful this can be. And finally, also to determine optimal doses of a drug. I'm not sure about, sorry, I'm just keeping track of time, so I'm hoping that you will stop me when I need to stop. As I said, these are all important and quite big challenges. And so the community, common disease that has been already very much a collaborative and collegiate community has now come together to fund what is called International Common Disease Alliance that really now already gathers hundreds of researchers worldwide. And this type of collaboration is working jointly to address these three problems of genetic mapping, of finding mechanism, and also developing medicine. But also importantly, beginning to design coordinated data infrastructure, as well as ethical protocols to drive, a more systematic and fairer approach to tackling common disease. And one of the things that we have been doing is to submit a fairly extensive white paper with some recommendations that really identify priorities that as a community we want to tackle. And we're beginning to design experiments that allow us to tackle these challenges in a systematic way. So in the last part of my talk, I just would like to tell you very briefly about the next stages in my career. So I was recently appointed by Technopole in Milano to lead or co-lead the Center of Genomics together with Piero Carninci, who's currently at the RIKEN in Japan. And really the idea here is that we want genomics to become a core component of the type of science that we do at Technopole. And so Piero and I will develop joint, coordinated programs, Piero focusing on functional genomics, and particularly the role of genetic variation and gene regulation, really at the molecular level, whereas I will continue to develop population and medical genomic approaches. So if you're not familiar with Technopole, really the overall mission is that to promote and contribute to improving human health and well-being. And this is done through, obviously, genomic and other high-tech approaches, and which aim to derive insights into personalized and preventative medicine that can improve human health. As part of the initial stages of the staff at the Institute, a lot of work has gone into setting up scientific facilities and services, which will be made available not only to Technopole scientists, but indeed also to external scientists throughout Italy. We obviously have a keenness to drive innovation and progress by developing technology transfer and in a way operate similar to other international institutes, disseminating scientific activities in Italy. So overall, the center, the Technopole has 5 centers. So as I said, the dual nature of the genomics center, as well as neurogenomics, structural biology, computational biology, and data science centers. And as we're beginning to populate the faculty portfolio of the Institute, we're also beginning to define in more detail some of the scientific projects that will be done there. So as I said, the Institute, which will be completed in 2025-26, has a state-of-the-art equipment, including very nice structural microscopy facility, a genomic facility, data centers, as well as an automated stem cell and organoid facility, with other being developed over time. We have headquarters in the Palazzo Italia, the Expo, which is now pretty much complete, and are beginning building temporary and permanent lab structures that will be ready some the middle of next year and some in 2025, 2026. So as I said, we really would like very much to welcome collaborations both locally and nationally, and we can think of doing that through, first of all, scientific collaboration, but also through operating the institute as an open hub, providing access to infrastructure, sharing expertise and training, providing access to crucial resources, as well as fostering and enhancing the ability to build large-scale strategic initiatives, such as the ones that I mentioned earlier. And very much, all of us are very, very happy to discuss opportunities for collaboration. And one of the first things that we set up is an early career fellowship program where we devote some of the funds to supporting development of talented early career investigators that are based in independent scientific institutions, but with strong strategic and collaborative links to our science. So what the Population Medical Genomics program will do, I think my scientific research has really presented what we are about and what we like to continue doing. We want to continue to do studies on genetics and genomics in health, disease, and human biology. And we're very particularly keen to harness some of the genetic diversity in the Italian population. We know Italian population is very diverse. So there is really a need to account for local structure and genetic diversity, not only for discovery, but also to enhance clinical interpretation. And in general, Southern European genomes are indeed quite underrepresented in global samples. And we like to set up studies and not only look at Italian genomes, but indeed the ability to access more diverse genomes and biosamples to gain genetic insights. So the faculty portfolio will work broadly on a number of topics, including population resources and genome structure, exemplar, common, and rare diseases to be defined, as well as I said, disease mechanism, particularly through in vitro studies, and beginning to explore some of these opportunities for translational genomics. And while the portfolio, the scientific portfolio will be determined in part by investigators hired, the common themes is that we really want to have genetics at the core of our science, use state-of-the-art genomic technologies, leverage quite heavily quantitative sciences and biostatistics, and also in vivo and in vitro models of disease. And so what we want to do is, in terms of population resources, is to develop national and international genomics initiatives, particularly to try and enhance understanding of genetic diversity and predisposition in Italy and in Southern Europe. We want to use high-read depth whole genome sequencing, as well as new sequencing generation technologies to generate comprehensive catalogs of genetic variations. In terms of common and rare diseases, really about genetic predisposition and how we can enhance that. What are the studies that we can do that are original in this space? For a mechanism, we want to really deploy high-throughput genomic analysis and in vitro experiments to decode key mechanisms underlying complex disease variants. And finally, what are the opportunities that we can do, particularly in collaboration with the local excellent clinical science in Italy to diagnose and treat human diseases. So one of the first studies that we're planning to do is to partner up with an excellent epidemiological cohort called Molisani, based in Molise, which has, over the course of, as the aim to evaluate risk and protective factors for chronic degenerative diseases, and particularly cancer, cardiovascular disease, and intermediate phenotypes. And over the course of the last 15 years, has recruited nearly 25,000 participants, randomized across GP records, and has collected a wealth of information, both of phenotypic information for disease risk factors, but also a very rich biobank of biological samples that we can access for some of the more detailed functional study. And finally, very good capture of different types of questionnaires And so, it's really the ability to tap into these underexploited from a genetic point of view resources that makes, I think, the appointment very, very exciting. So in summary, really, I hope I gave you the message that genomics provides powerful tools to unravel the causes, mechanisms, and treatments of disease. There's still a lot of open challenges in terms of how we can harness the wealth of genomic data in the clinic. And it is very clear that confederate and systematic approaches to this type of science are indeed very needed in the community. So with this, I'd like to thank everybody. So this is my team. And many, many collaborators that I couldn't list here, but they are in the hundreds. And really thank you for attention, and also tell you that there are obviously positions, and we always welcome contact by people that might be interested in working for us or with us at the Technopole. Thank you.